So have you all completed your sign up on Learn?
Yes.
Okay, that's great. And I see a lot many new participants as well today. So I'll take you through how you can sign up on Learn and get access to the asynchronous material, asynchronous course material, and leverage all the benefits. Now I see Ali Hassan, you are from a research fellowship program. Have you completed your profile? You can unmute yourself. Hi, Mithali. Are you from India?
Yes, ma'am. I'm from India.
That's great. What are you pursuing right now?
I'm doing MSc Biochemistry in Reva University.
Okay, you are from Reva University. And which year are you in?
First year, ma'am. First year
Okay.
MSc.
Okay, and what are your research interests?
Ma'am, just someone suggested me. Today is my first. Today is my first class, ma'am.
Okay,
Hi,
fine. So you
Mithali.
want to learn.
So
Yes,
yeah, it's good to have you. Do you know Dr. Ramesh from your
yes,
department?
sir.
Yeah.
Yes, sir.
Okay, so we have had a data science program for 1 month for your seniors, perhaps second year students. So many, I think 50 people or plus 60 people did that course, and also have interns from your university who are working with us, Shreyashi and Shreyoshi. I think if you know them. All right, thank you for joining today's meeting, and we'll be starting in a minute or so. And yeah, over to you, Sonalika.
Sonalika, we can't hear you. Sorry. Sonalika?
Sonalika, you're muted. Can you see us?
But yes, actually, I'm joined from another device with my mic on. Can you hear me?
Now we can hear you. Can you start over again, please?
But we can't see you either.
Okay, fine. Actually, I'm connected from two systems right now because of some issue. So yes, just give me a second. So I was saying that we have a lot many new participants as well today. And that's why I would be introducing ourselves first, and then we'll take over the presentations for today's research fellowship meeting. Introducing myself, I'm Sonalika Ray, and I'm the OmicsLogic Community Manager, and I'm responsible for daily interaction with thousands of users that are a part of a bioinformatics community, which is managed by Pine Biotech. Introducing our company, OmicsLogic is an international program that is running in 5 different regions with over 10,000 users around the world. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources as well, adapting them to the needs of students and researchers around the world. So for those joining for the first time, We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. Talking about the Bioinformatics Research Fellowship Program. Yes, I'm so sorry. Talking about the Bioinformatics Research Fellowship Program, it is a very structured program that guides students through various areas of big data bioinformatics research using practical examples. And during this program, we would go through several high-quality research publications and learn about applications of computational biology in projects these publications describe. And this kind of an experience allows beginners to try computational biology techniques on public domain data, making it possible to work with large files and extract meaningful information from patient samples, animal models, cell lines, and microbiota. Our training is designed to allow participants to apply methods to curated and new data sets and develop independent projects that showcase what has been learned in the context of a personal portfolio of projects. And as a result, anyone starting on the journey in bioinformatics can start developing a project of their own. In fact, we have already worked with many students who have completed this kind of training and developed interesting research projects that won awards and got published in peer-reviewed journals. So before we begin with today's research fellowship presentations, let us quickly go to learn.OmicsLogic. As I can see, we have many new participants today. So those who are new here, I would want them to go to learn.OmicsLogic. Let me know once you are here. I'm sending the link in the chat box. Once you visit learn.OmicsLogic portal, you need to sign up on the portal, and you can do so with either your by creating your ID and password, or by signing up with any of your social accounts. So those who have joined the meeting for the first time, once you are signed up, please let me know in the chat box by sending either 1 or yes, so that I know you have signed up. Signing up is
maybe Sonalika, I could also clarify for those that are signing up. And especially I want to make this clear for those that are a part of the LBRN program. We need your ID once you sign up. So let me share with you what is the ID. Maybe Sonalika, can I share my screen?
Yes, sure.
Okay. Okay, so once you sign up, so again, if you're not signed up yet, you can go to log in. And on the login screen, there will be an option to sign up. Okay, actually, I'm going to stop sharing this 1 second until this loads. Okay, so what we need for our records, the reason that we are doing this specifically for the LBRN is because you have a scholarship from LBRN. So right here, you will have create an account. And so Monica, please paste this link directly in the chat for the sign up. So once you create an account and you log in, let me just briefly log in here. What we need from you is the link to your profile. And that is a specific code. You can get that code by clicking right here on your name. And this right here on top is the full URL. That's what we need. So please send that to us, either paste it in the chat, or you can send it to us by email. Okay, that's an example. So you can go ahead, Sonika.
Thank you so much, Ilya. So we have, wait a second. Yes. Hi, Ali. Can you? I would want the participants to mute themselves. I can't see who is unmuted. Yes, thank you so much. Hi, Ali. So starting with you, we would want to know about your research progress.
Hello, ma'am. Good evening.
Hi, Ali. Good evening.
I was very sick. I was infected with COVID. That's why I didn't do that much progress in my study. I think I need more time in this week.
Okay.
I assure you, I will not disappoint you.
Right. No issues at all. Thank you, Ali. Yes. Now I would want Dr. Harpreet to please introduce herself to the new participants we have for the research fellowship program, and then we may proceed with the presentations. Over to you, Dr. Harpreet.
Thank you, Sonalika. Let me just share my screen. Hello, everyone. My name is Harpreet Kaur, and I have a PhD in bioinformatics. But before my PhD, I have done my master's in molecular biology and biochemistry. So after doing my master's, I was always interested in learning like how these molecular mechanisms playing important role in different disease conditions. And besides, I also know that there is continuously enormous NGS data is generated by number of the research labs for thousands of the patients. So I believe one can analyze such data to understand what are the biological and molecular processes that are taking place underlying specific disease condition. So I have joined PhD in bioinformatics at CSIR Institute of Microbial Technology, Chandigarh in India, under the Professor GPS Raghwa's lab. So my PhD work on biomarker discovery for liver cancer. So during my PhD, first, why I chosen this topic? So it is well known that liver cancer is one of the major lethal malignancy, which is considered as third major liver malignancy in terms of mortality, while sixth in terms of number of incidents. So then I went through the literature to understand what are the challenges that are associated with the liver cancer. So during this my literature review process, I have found there is a need to identify important signatures that can predict liver cancer at an early stage. And there is still need another factor that contributes to the poor prognosis of the liver cancer, that is tumor reoccurrence. So based on my literature review process, I understand that there is a major concern that needs to be addressed. That is, first is accurate cancer prediction, particularly for the liver cancer, both hepatocellular carcinoma and cholangiocarcinoma. Second is its diagnosis at an early stage. Then there is a need to identify important signature that can stratify HCC patients into low and high risk patients. So based on this literature background, first we generated a resource, that is cancer liver, where we compiled the data associated with nearly 600 biomarkers to understand what are the major limitations associated with already known signatures and the biomarker. In the second part of this cancer liver resource, we also compiled the data for nearly 10,000 liver cancer patients from different public repositories so that we can address those concerns. So in the next project, we have extracted the data from different public repositories, including GEO and TCGA, for nearly 4,000 patients to identify a robust diagnostic biomarker that can predict hepatocellular carcinoma with maximum accuracy. So in the subsequent project, here we have extracted the data from the TCGA for nearly 350 patients. And here we have tried to identify signatures based on transcriptomics and epigenomics data that can classify early and the late stage liver cancer samples with nearly accuracy 80%. So then we focused on the next malignancy for the liver cancer, that is cholangiocarcinoma. So to identify robust transcriptomic signature for the cholangiocarcinoma, we have again extracted the data from the GEO and other public repositories for the cholangiocarcinoma. So here we have extracted nearly 600 samples, and then we have tried to identify important signature based on the 3 genes that can predict cholangiocarcinoma or classify cholangiocarcinoma from HCC in the normal sample with nearly 90% accuracy. So then eventually, based on all my during my PhD work, first we have identified 3 gene-based signature that can predict HCC from the normal sample with more than 90% accuracy. And in the second part, we have identified 3 gene-based signatures, that is LAMC2, ITZA, and C2, that can discriminate cholangiocarcinoma for normal and HCC sample with nearly 90% accuracy. And in the next part, we have identified 51 features that include 30 RNA transcript and 21 methylation CPD sites that can classify early stage samples from the late stage sample with nearly 80% accuracy. In the last part, we also try to predict some signatures that can stratify low and high risk reoccurrence groups of the SCC patients. So apart from that, during my doctoral research project, I have co-mentored 4 research fellows who are actually MTech students. And in one of the projects, we have created a resource that is Rare LSD, which is a manually curated database for the lysosomal enzyme associated with the rare disease. And in the second part, we have created a database for the biomarkers for the prostate cancer. And both of these works get published, and two other works are still in communication. So apart from that, when I joined Pine Biotech, here I have mentored number of the research fellows. So the first, we have Simay Dolanar, who is an undergraduate student at Turkey, and her work is already published, where she have worked on identifying the long non-coding RNA as therapeutic target and in chronic lymphocytic leukemia. And she have used the publicly available data for the CLL cancer, and she implemented various bioinformatics tools that are integrated on T1 for server. And in the another project, Urja Parekh, she's also completed her research project entitled In Silico Analysis of Characterization of Differentially Expressed Gene to Distinguish Glioma Stem Cells from the normal neural stem cells using publicly available data from the NCBI itself by implementing various in silico tools that are integrated on T1 for server. So then we have Mr. Kalamari, who is a PhD student at Mexico, and he has worked on a project classification of triple negative breast cancer cell line by gene expression analysis of transmembrane protein implementing machine learning techniques. Apart from them, then we have Dilara Duquesne, who worked on metagenomics project entitled Radiation 3 to the Human in Space, an Alternative Approach with the Probiotics. So apart from graduate and postgraduate students, then we have high school students, and one of them is Netra Srinivasan, who worked on a research project entitled Effect of the Onset of Menopause on the Transcriptomic Profile of the Aging Female. And she has also taken the data from the public repository, that is GEO, and she implemented various bioinformatics tools to identify important transcriptomic signatures. Then we have another high school student, Mr. Andres Sosa. So he also worked on Alzheimer disease, where he used publicly available data to identify amyloid beta induced neutrophil activation that may provide a link between two pathological hallmarks for the Alzheimer's disease. So I'm looking forward being your mentor. So thank you, everyone.
Thank you, Dr. Harpreet. And thank you everyone again for joining. So let me kind of pick it up from where Dr. Harpreet finished. She mentioned some of these projects, and as you can see, the projects come from very different backgrounds. Some people are from high school level, some people are from undergraduate or even graduate PhD students. And I think that's what's important about this program. Today in the session, you can see the diverse backgrounds of everybody who's participating. We have here people that are from around the world, and all that connects us is this, I think, passion for research and our interest in bioinformatics, but also there are many differences, right? So there's many different interests and many different directions that you will be taking throughout your research fellowship. So let me briefly describe how this research fellowship is going to work. The place to start is to complete your profile. Really, that is where you're going to have all of the information about you so that we can see it, your mentor can see it, and others will see when they look at your project. So again, where is your profile? Your profile is, if you click right here, and what's important is to have a profile picture and social media links, because we want to know not just who you are, but also connect with you. And we want to encourage this networking between the research fellows as well. So typically your professional profile is going to be on LinkedIn. Maybe your research profile is going to be right here on OrcID and others. And you can also write a brief description about yourself. You can edit all of this information right here. Now, then down below, you will see some points. These are very important points that we can keep track of your progress, but also you can know where you are and how consistent you are in your learning. So as you join the research fellowship, start by looking at existing materials that we have here that will give you a sense of the different types of data that you can work with, how to find this data, some project examples that you can reference in your study, etc. Then you will have right here your subscription. So all of you that are already a part of the research fellowship should have a starter subscription that gives you access to all of the premium materials on the portal. If you don't have this, please do let us know. The easiest would be to let us know right now. If you don't see that, you just write it in chat and we will check it for you and make sure that you have it before the end of the session. Also, you will find down below some badges, and these badges correlate to the different programs that we have. These programs include training on data science, infectious diseases, precision medicine, SARS-CoV-2, and others. And so if you want to learn about these, you can explore the different types of programs that we have by going right here on the programs tab and finding here a mentor guided program that is most suitable for you. Okay, so Nalika, please take a look. Yushi Gaur is saying that she doesn't see the starter subscription, so we need to fix that. Yushi, if you can please post the URL to your profile, that will help us find you quickly. Okay, so what is the URL to your profile? Right here. That full URL is what you need to put in the chat. Does anyone else have any issues with their subscription? Thank you. Okay, so we can go to this profile. So again, please try to keep your profile with the full name as you would like it to appear on your certificate. Okay, so eliminate this 01. Please have a full name. Please add your picture, social media links, and a brief intro, and we will make sure that we have your information updated and give you the full subscription. So Nanika, it also looks like Gurpreet Bamra does not have the full subscription either.
Oh, so I would like to. Okay, I'm so sorry for this. Yes. So I would like to tell here that to get the access to our start content, you need to take the basic subscription that is of $10. And when I say that you can sign up for free, that means that you would have the access to all the asynchronous course material that is present for free on our portal. But all the start content could be accessed only with the basic subscription that you would have to proceed with. I'll send you the details, Ayushi. And I just checked your subscription is not active just because you don't have the basic subscription activated for yourself. And Gurpreet, Ayushi, please tell me your mail IDs in the chat box itself. I'll mail you the details. Thank you. Over to you, Ilya.
Thank you. Yeah. So again, if you want to join the research fellowship, you haven't finalized that yet, you can just click right here to research and you can find the different options. If you're already a part of LBRN and your subscription has been supported by the Louisiana Biomedical Research Network, you need to let us know. And especially if you don't have the subscriptions finalized. Okay, so now because we do have a lot of new people joining today, maybe we can hear briefly about you, why you're joining the research fellowship, what you are seeking to gain out of this experience, maybe what is your current background. So please let me start with Abu Bakr Abdul Qadir. Abu Bakr, if you could please introduce yourself.
Hello.
Hello.
May I get a speech problem again? Sorry.
Yes, Abu Bakr, you can tell us about yourself in the chat box. Thank you so much. Then we have Ariana with us. Ariana, can you please introduce yourself?
Sonanika, one second. I think Abu Bakr, his microphone is working. Abu Bakr, are you able to introduce yourself?
So Ilya, I think Abu Bakr is trying to say that he has some speech issue and he can't speak. So he wants to introduce himself in the chat box. Yes. Thank you so much.
Well,
so we have
he did
Ariana.
write before. I see. So he did write before. Let's just read that out real quick. So he's saying that he's a PhD student in environmental toxicology at Southern University A&M College Baton Rouge. Okay, and he's working on the immunotoxicological effects of some toxics. So if you could add to this information, this is good information, Abu Bakr, thank you. If you could add to this the information about your the reason why you joined. So what are you seeking to get out of this program? Are you do you have a specific project that you're planning to work on? Do you already have a project in mind, a topic or a research question? What do you envision getting out of this 3 months fellowship? Thank you. Please go ahead, Sonanika. Who else do we have?
Thank you, Ilya. We have Ariana with us today. She recently joined a research fellowship program. So Ariana, I would want you to please introduce yourself first. Over to you, and also tell us about your research interests and what you're looking forward to. Over to you, Ariana.
Hello, hey, Ariana is my daughter. She is taking a class now. We are in Arizona, Phoenix, Arizona. She is a high school student, but she is the summer class in Maricopa Community College. And I saw that we were talking to Dr. Tui in the Glendale Community College, where I see you guys have taught a class with him in bioinformatics last semester. So that's where we get kind of a connection. So Ariana is taking the bio class right now, bio 181 with the community college. So she is not able to join, and she asked me to talk to you guys here. Now, her interest is she's doing in the community college, taking some bio classes, and at the same time, she is doing her artificial intelligence program in the community college here, Chandler Gilbert Community College. So she has some idea, she has some background on the Python, she's doing some machine learning courses. And then she is also pursuing the biology. So she has some interest around that. She's only an 8th grade she's only 9th grade student. She has interest in doing some research projects so that she can turn into a science fair project through the research. So that is her goal, science fair project. So she hasn't chosen anything yet. She is looking for some mentorship guidance. As I said, she is a very young, only 14 years old. So some guidance would be very good for her. We are seeking some help if this is a possibility. And then, I mean, she is very aggressive. She wants to learn more. So whenever there's an opportunity, we wanted to sync up with you guys and see what are the options available, especially learning the data sources and and what is the popular thing needs to be researched in this current moment, and what is the pretty popular so that she can turn into a science fair. That is it. Thank you. If you guys have any comments, let us know.
I do have a question. So I did understand that you're the father of Ariana. What is your name?
Mizan.
Mazan. And has Ariana already created a profile?
I think she did. Yes. Yeah. I can send it to you here. I believe she did, but I don't think she has updated. Yeah.
Okay. Perfect. Ariana Rahman. All right. Great. Sounds good. Yes. So we will try to find a time that works also for her. We do have a batch of advanced high school students in the US that are going to be meeting in the afternoons. So maybe that is one of the batches that we could include her in.
That'd be better. Yeah. Sure. Yeah. And then are you guys offering any class, any more like bioinformatics class with the Glendale Community College with the Dr. Tui in next semester?
Yes, I think we're planning to, but we need to coordinate with him exactly how that will work and what are the dates. So that will be more up to him, but I think we're planning to speak with him later this week.
Okay. So Ariana, yeah, we are also meeting him tomorrow, and Ariana will be in his lab. So she will be taking some classes under him and then as a research part of the, also probably 1 or 2 credit under him so that she can have access to the lab.
Perfect. Okay.
Awesome.
Great to know. Thank you.
Yep.
Thank you so much, sir. And thank you so much, Ilya. Now I would want our research fellow, Rucha Koneti, to please present her research progress. Rucha, I'm giving you the access to share your screen. Well, Dr. Mohit, can you please give her the access? Make her the co-host.
Okay.
Thank you.
Hello, can you see my screen?
Yes, we can see your screen. We can hear you as well. Thank you, Rucha.
Okay, so today I'll be giving my progress in my project, transcriptomic analysis of neurons and oligodendrocytes of the occipital lobe in Alzheimer's disease. So first of all, let me introduce myself. My name is Rucha and I'm from Bangalore, India. I'm currently studying biotechnology at Ramaya Institute of Technology. So I've previously done molecular docking studies on drug-resistant tuberculosis and SARS-CoV-2. But my interest always was in genetics, genomics, epigenetics, and transcriptomics, and how they can be used to analyze various diseases. So I'm also interested in learning about, the various illnesses that affect the brain, like neurodegenerative diseases or personality disorders like ADHD, bipolar disorders, etc. So that's why I chose to do my project on Alzheimer's disease, which is a neurodegenerative disorder, and it affects individuals of over 60 years of age or people with trisomy 21. And common symptoms include memory and cognitive impairments, And with further disease progression, we have other symptoms like delusions, agitations, and mood disturbances also. And AD is mostly characterized by the formation of protein aggregates, that is neuritic plaques and neurofibrillary tangles. These plaques are actually the beta amyloid peptide aggregates, and the tangles are actually caused due to the hyperphosphorylation of the tau protein. So you can see here, these are the tangles and these are the plaques. Um, so, one second. Okay, so so most studies have established that microglia, which are the resident macrophages in the brain, these are involved in the AD pathology, and the neurons in the limbic regions, that is the hippocampus, temporal lobe, and the parietal lobe of the brain, are affected. And we know that neurons are the fundamental units of the CNS, so they're involved in impulse transmission in the body. So now research is still being done to find out if other cells and tissues of the brain are affected, like oligodendrocytes or blood vessels, etc. So now oligodendrocytes are a type of glial cells which are non-neuronal cells, and they don't conduct impulses, but they're involved in homeostasis, and they provide support and protection to the neurons. So oligodendrocytes in particular, they are involved in formation of the myelin sheath around the axons. So now what is the occipital lobe? So this region is present at the posterior region of the brain, and it includes regions like occipital cortex and occipitotemporal cortex, and is involved in visual perception, spatial reasoning, and processes input from other regions of the brain. So now scientists are still researching the full effects of AD, like on different parts of the brain, not just the limbic system. So that is why yeah, so that is why I chose to do my project on what type of cells are affected, and in this particular region in Alzheimer's disease. So for this project, I chose my data set from this bio project, geo data set, which actually analyzed the microglia profiles in AD. So in this data set, there are cell samples obtained from the occipital lobe of the brain from 3 age-matched groups, that is AD, control, and control plus. So the AD samples, they have both the amyloid beta pathology and the tau pathology. And the control samples have neither amyloid beta nor tau pathology. And control plus samples have some level of amyloid pathology, but there's no tau protein aggregation. So, and the difference between these control and AD is that AD brains are they have signature dementia, while the control samples, they are non-demented brains. So this is the data set, and there are total 59 samples, and 26 of them are oligodendrocytes, and 33 are neuronal samples. And these cells are obtained only from the gray matter region. So so my research questions and hypotheses are, so we know that memory and cognitive impairments are the major symptoms of AD, and these functions are under the control of the limbic system. So are neurons of only these regions being affected, or neurons of other brain regions are also being affected in the same way in AD? That's my question. And also in addition, like what other cells are being affected, like oligodendrocytes, and again oligodendrocytes of the occipital lobe in this case. And if so, what genes are being upregulated or downregulated in the affected cells? And what are the pathways and biological functions being compromised due to AD in these cells? So I first did a principal component analysis for the cell samples. And you can see this is PC1 versus PC3. And these red clusters are neuronal samples, and the blue ones are the oligodendrocytes. So this contributes to a total of 70% variance in the whole data set. So then I did a differential gene expression analysis of 8,481 genes using DC2, and with p-value of less than or p-value less than or equal to 0.05. So I did 4 types of DG analysis, 2 for neurons and 2 for oligodendrocytes. And these are the results I got. So in the neuronal cell sample, in AD versus CTR, I got 34 differentially expressed genes. And in AD versus control plus, I got 168 differentially expressed genes. Similarly, in oligodendrocytes, I got 399 for AD versus CTR and 56 for AD versus control plus. So today I'll be talking about these results in particular. So in the differentially expressed genes of the neurons, so I annotated them according to gene ontology and found that there were no major role in played by these genes in AD related changes. So which probably explains why this PCA looks like this. All the samples are like present together. They don't form separate clusters. So that's probably why. And this is the same neuronal samples, just the groups have been separated. This is AD versus CTR, and this is AD versus CTR plus. You can see that there's no clear cluster formation. Coming to oligodendrocytes, I again did the same thing. I annotated them according to gene ontology and found that many of the genes were playing a role in AD progression. So you can see here, the clusters are a bit more clear. These are the AD samples, and these are the control. The control plus is kind of present in between, probably because they have characteristics of both. Like they are similar to control as their non-demented brains, and they're also similar to the AD samples because they have the amyloid beta plaques. So going further into the oligodendrocytes, this is the same thing, the PCA for AD versus CTR and AD versus control plus. There's a clear cluster formation here. So going further for the oligodendrocytes, first I'll be talking about the AD versus control plus. So here among the upregulated genes, I found GLI3, which is actually involved in the WNT pathway and causes its negative regulation, which contributes to the loss of impulse conduction in AD patients. So WNT pathway is actually impulse conduction pathway, broadly speaking, and this is very important in maintaining the synaptic plasticity in the adult brains. And among the downregulated genes in this group, I found that there are a lot of mitochondrial genes being downregulated, which cause the disruption in the electron transport system, leading to oxidative stress, which actually further induces the amyloid beta growth in the cells. So this is a volcano plot of oligodendrocytes AD versus CTR, and you can see the mitochondrial genes being downregulated here. And next is AD versus CTR. So here I found some genes like protein kinase, genes encoding protein kinases like AK5 and PRGK1 being upregulated. So these kinases are actually involved in phosphorylating proteins and are involved in microtubule formation. So AK5 is pretty important in maintaining the microtubule structure. So since it's being upregulated, it actually hyperphosphorylates the tau protein. The tau protein is actually required for proper microtubule formation, and if it gets phosphorylated to a greater extent, it can negatively regulate the MT assembly, which is actually what happens in the AD patients, and that's why microtubules are very weak in AD patients. So you can see here AK5 is being upregulated. This is a volcano plot for AD versus CTR oligodendrocyte samples. Next, coming to the downregulated genes in AD versus CTR, I found these two genes being downregulated, that is BACE2 and APC2. So APC2 is a WNT signaling pathway regulator and is also involved in the microtubule formation. So like I said, it's very, the WNT signaling pathway is pretty important for maintaining synaptic plasticity in the adult brain. So since it's being downregulated, that is why we have a synapse dysfunction in AD patients. And BACE2 is a beta secretase 2 enzyme, which is actually homologous to the main beta secretase enzyme BACE1. Now, BACE1 is what actually causes the amyloid beta plaque progression in AD patients. It happens when BACE1 gets upregulated. So, and at the same time, BACE2 is being downregulated. So that's what adds to the amyloid beta progression. So BACE2 actually in healthy individuals is expressed in relatively lower concentrations, but it does play a pretty important role in delaying AD progression. This is because it cleaves the APP, which is actually which is nothing but the amyloid precursor protein. And as the name says, it's the precursor protein for the amyloid beta aggregates. So it cleaves APP at a particular site different from the ACE1 site, and that's how it prevents the amyloid beta progression. So since it's being downregulated here, it's probably why the amyloid beta plaques progress in AD in addition to the upregulation of the ACE1. So, and these are some of the enriched pathways I found when I did DC2 analysis. So you can see here in neurons, you can see here there's not much of genes involved in AD progression, whereas here you can see a lot of mitochondrial functions being suppressed in the oligodendrocytes AD versus control plus. And here in the neurons, again, you don't see much of AD related changes, whereas here you can see a lot of nerve impulse conduction and neurotransmitter activity being suppressed. So from this, it seems that neurons are not as affected as neurons of the limbic system as one would have thought. Like you'd think neurons of all the brain regions would be affected, but this is not so. So in the occipital lobe, neurons seem relatively unaffected compared to the oligodendrocytes. So with this, I end today's presentation, and after this, I thought I could do some pathway analysis, like why this difference is seen, why neurons are relatively unaffected than the oligodendrocytes, which I thought I'll do in the coming week. So thank you everyone for listening.
Thank you so much, Rucha, for such a wonderful presentation. I would want Dr. Harpreet to please give the suggestions now.
Okay, so Rucha, I mean, you did a lot of analysis, but somewhere you need to interpret it a better way. So can you just go to the your volcano plots? Actually, you are just, I mean, confusing here yourself here. Okay, so can you
Oh.
just show the plot? Yeah. So actually the genes that, can you tell me, like, based on the log2 fold chains, which genes can be considered as upregulated and which genes can be considered as downregulated?
It's the log2 fold chain positive values are upregulated, but negative values are downregulated.
Exactly. Now can you look at your plot, volcano plot?
Yes.
So
Ma'am,
these genes
this
are upregulated that are present on right side, and the genes that are present on the left side, because here -4, -2, right? If you
yes,
look
ma'am.
at the,
I understand that, but I'm
yeah, but maybe you are talking about in terms of when you actually decide your groups, group 1 and group 2. If you remember, when you
yes,
run your
ma'am.
DSEC, from that perspective, you have to mention like that. Actually, that is why I'm saying you are you are doing wrong interpretation in terms of upregulation or the downregulation, because then you are talking about in terms of which group you are talking about, right? You have to
Yes, ma'am.
consider that. So based on this, like the positive log2 fold chains means these are upregulated genes, while negative fold chains means these are downregulated genes. So this is something you need to correct. And
One second, ma'am. That is true in case of control plus. I was talking in terms of AD, ma'am.
yeah, I mean, then you have to mention exact group when you're
Okay,
creating
yeah, I
actually
forgot to
upregulated.
mention. Yes, ma'am. I'm sorry. I'm sorry
Because
about
you
that.
have to write, because we are looking at this result. So based on this
Yes, ma'am.
result, we are going to interpret. So you have to mention in which context you are talking about, right?
Okay, okay. I'm sorry. I forgot to mention that. Yes, ma'am.
Because based on this plot, you can say that this particular genes are upregulated in whatever the condition. I mean, you have to mention that condition here,
Okay,
right?
okay. Yes, ma'am.
Okay. So now I would ask Ilya to if he give you some suggestions.
Yeah, thank you. It was very interesting to hear the presentation. As far as I understood, your main question is development of Alzheimer's disease, or really the manifestation of clinical symptoms of Alzheimer's disease in specific types of cells, neurons versus oligo, whatever they're called, oligodendrocytes,
Yeah.
right?
Yes, yes.
And so you're trying to ask this question, and my question would be, is this really a question that has not been answered yet? Is there literature examples that show that this is a very important topic to study? It has not been studied before?
Yeah, I researched a lot about this, and there are mostly research articles only based on the neurons of the limbic system. So other cells, there's not much research done at all yet. So it's still going on.
Okay. Doing a search, I do see some articles that speak about this. Early oligodendrocyte myelin pathology in Alzheimer's, generation of human neurons and oligodendrocytes, neuro-oligodendrocyte interactions, right? So it seems like there has been some research already done on this
Yeah, but
topic.
it's very less compared to the actual what people are finding now.
Neurons. So
Yeah.
you're saying that so that's what I think you should make clear in your specific objectives, is how what you are proposing to do, or what you're already doing, how is that going to address some of these gaps in research that you see?
Okay.
Okay, and also, what is the utility? So let's say you understand this. Let's say you get to the bottom of activated pathways, specifically in oligodendrocytes.
Yeah.
How is this important? What are we going to do with this information?
The fact that some of these neurons were unaffected, but these oligodendrocytes were affected, maybe there's some pathways being like some channels like cation channels or something being affected. So maybe that's what's protecting those neurons, and analyzing those particular pathways can help us like find ways to protect all the other cells.
Okay. So maybe you should refine that idea also. What exactly is the, so have you ever looked at cell morphology, what they look like?
What neurons look like?
Yeah, well, both of these. I think if you go back, you had a slide, right?
Yeah, it's here.
Yeah.
These are neurons, and this is oligodendrocytes.
So, I mean, we can see that they're already very different, right?
Yeah.
And because they're very different, we are expecting them to have differences even in healthy tissue, right?
Yes, that's true.
And from what I've seen, I have not seen a comparison between Alzheimer's. So if you have a sense of difference between oligodendrocytes in Alzheimer's disease and non-Alzheimer's disease, as well as neuron cells in Alzheimer's versus non-Alzheimer's, you could have then a comparison of differentially expressed genes in Alzheimer's between oligodendrocytes and neurons, right?
Okay, yeah.
And that could be an interesting question. If that's really the focus of your study, you could then see whether there are specific genes or even pathways, and what does that mean? And so as a result, actually, you can turn this whole study into kind of the next level, which is to do a classification of 4 different groups. So you have oligodendrocytes, Alzheimer's, non-Alzheimer's, neurons,
Yes.
Alzheimer's, non-Alzheimer's. So you have 4 classes. So it would be interesting to do classification and feature selection to extract specific genes that could help you identify what are the, what are the markers of these conditions. And when you have that, there's a single cell atlas of Alzheimer's disease.
Okay.
Where you could just look up those genes, and that would be a cross-reference or kind of a validation study that you could do to check whether you find those genes and they are regulated in a specific way in different tissues.
Okay. So
Well,
I'm to do a feature selection for all the 4 groups. Is that what you're saying?
Yeah.
Okay.
How many total number of samples do you have again? I think you have a slide on that.
59. Total, it's 59.
So maybe it's not enough in this study, but
Yeah,
you can definitely.
It's also considering only the gray matter area, so so maybe.
But you can still think about it. I think that would be a useful approach, because if you do differential gene expression between many to many groups, you will get some genes. And the typical way to then check those genes is to find another data set. And so maybe you can take a look at, if you have maybe over 100 samples, you could do something with classification and feature selection. That will give you some sense of, what is the utility of this? I think the utility in terms of basic biology, if you're trying to understand activated pathways, you need to then take that, into the lab and start looking at whether you can do something with that. As opposed to a classification approach, the classification approach can give you some markers, and those markers could help you identify, for example, in mouse tissue, right? In human tissue, maybe that's not possible, but in mouse tissue, you could ask whether those genes have some variation in age, whether they have some variation with inflammation caused by other factors not related to age, right? So there's some additional questions that could make your study objective more clear, because right now it sounds like you are investigating this to understand the biology, but I don't think that necessarily translates to protection if you identify those pathways. Or if it does, then I think we need to clarify how can that be. You know what I'm saying?
Yeah. Okay. So I thought of finding the reason why it's happening like this and then seeing, is that okay?
Well, yeah, maybe you should definitely start with literature review. Go back and see what are people saying about why these kinds of studies need to be done and what are the unanswered questions specifically and why it is important to answer these questions. And then from all the possible ideas that you get from literature review, select one focus for you. And then from there, decide whether what you are currently doing is sufficient for you to get some activated pathways. What are you going to do with that information? And if you have specific genes, what, if you have, let's say, 500 differentially expressed genes, it's not very useful to look at just 500 genes, right? So you need a smaller number. And the smaller number means that you have to put the 500 genes into some context. For example, you can do network analysis and identify key genes. You can do classification to extract genes that are very specific and help you discriminate between the different classes. Then you might look for those in maybe blood, and maybe there's some circulating biomarkers for detection that you could find, right? So there's a lot of then downstream questions that we need to refine as well. But, I don't want to take away from the significance of what you already did. I think it's an excellent way to get started and get into the topic. You already have a lot of results. It's a useful starting point. Maybe with just more clarification, it's a study on its own.
Okay. I'll think about it and we'll do it.
Perfect. Thank you.
Thank you.
Thank you so much, Rucha, and thank you so much, Ilya. Now I would want Hriday, our research fellow, to please share his progress. Over to you, Hriday.
Sure, ma'am. Due to bandwidth issues, I won't be able to turn on the video. And if it's all right with everyone, can I start the PPT?
Yes, you can. It's perfectly fine. Thank you, Hriday.
Okay. Is the screen visible? If I can ask everyone.
Yes, Hriday, your screen is visible.
All right. Perfect. So thank you everyone, first of all, who are present here in the meeting. I am Hriday Thakur. I have been a research fellow with Pine Biotech, and my research is going to be on a topic that is of concern for many of the people in this world, and that is prostate cancer. Now, the prostate cancer disease, it's a very common disease, and it's found mostly in aged males. And most of the times, from the literature that I found out, that the common thing that was present is that was mostly found in African American males and very aged males. So this is how I started to develop interest in this topic, and the tools available from Pine Biotech really helped me to analyze this. I'm still on the very preliminary step, but this is what I did, and just to show you and present what I did in my topic. So what we first understood was about RNA-seq and NGS sequencing, what it really is. So when we talk about transcriptomics, we did a course on it, and it was very exhaustive and very interesting to understand what transcriptomics is, having no background of this topic and this subject. So we understood what finally transcriptomics is, what transcriptome is, and we understood all the bases of it, from RNA sequencing to NGS sequencing, how the sequencing developed. Finally, we understood that what the gene expression studies were and how total RNA was cut, then segmented, analyzed using the reference genome, then plotted, and then understood. So when we understood about next generation sequencing, about all the things that next generation sequencing did, with isoforms generation and alternate splicing, everything related to it when a library was created. So when we did this, we understood more about short read sequencing, long reads that were available. As you can see in the slide, it says that short read sequencing is actually cheaper, and which is very big advantage in the real world when we have to apply it in the economies which are not really very well done into research and development for genomics. And we can see that long reads are facilitate the analysis of challenging genomes that are not being studied yet. And these genomes with a high percentage of repetitive elements can be studied using the long reads. So this is what we finally understood, and we understood that what actually next generation sequencing can really help us in. When we understood this fact from, we came a long way from northern blotting and RT-PCR to finally come to this point, and microarray to finally reach this point where we can actually sequence entire genomes. So moving on, these are the basic and broad steps of how RNA sequencing works. So first of all is the pre-processing steps that takes place in RNA-seq, and we understand that these are first utilized using the FASTA and FASTQ formats, which are the formats in which the genomic data is prescribed into. Then we align these reads, we map them into a reference genome, and finally calculating the abundance of the reads along the reference genome, which tells us how much and what is the Q score associated with it. Finally, and the last process of the step would be to statistically and biologically interpretation of the expression table. And this is what this is what our end goal is in this project as well, to understand prostate cancer and the expression levels of different biomarkers in prostate cancer. So this is what our final result is going to be. And so moving on, we have this first step of pre-processing the data. So first of all, using pre-processing, it can help us eliminate unwanted technical artifacts such as the adopters that were present. And sequencing adopters are cleaned from the reads, and on the T-Bioinfo platform, there are 5 algorithms that are present for pre-processing. That is clean primer, array to FASTA, PCR clean, chemomatic. That is the one we use. And then there was GTF adjust. Error correction can be done with the help of Aimer, Coral, Echo, and Racer. And these are also available. About 75% of all sequencing errors can be corrected through these algorithms. And this is the most important part, because if the bases are not correct, then it's going to be a very tough job for everyone to analyze it. So finally, the sequencing adopters, they need to be cleaned from the reads, and the raw read data needs to be converted into a format that can be efficiently used for the subsequent algorithms to work on. And then we come to the second step, that would be to mark it and the map on reference genome. So the raw transcriptomic reads are aligned on the genome sequence for the same or similar species, and on this reference genome. This helps us to actually quantify the reads associated with the expressed genes. So when we talk about this mapping, we have many algorithms that are present on the T-Bioinfo platform, that are top hat, bow tie, and we can also do isoform construction using cufflinks, and splice variants are generated using the information read, alignment with the exons and introns yielded from the above top hat and high set that were algorithms that were present. So this is what we do we did on mapping and reference genome, and we understood about it. The next step would be to calculate the abundance of reads. Now, from what I understood was that there are 2 main formulas that are present. First is actually the reads that are read count that are present, and the next evolved formulas are the FPKM and the RPKM values. Both of the have been elaborated in the slide. The RPKM stands for reads per kilobase of transcripts per million mapped reads, and FPKM would go for then fragments per kilobase of transcript per million mapped reads. And so these are the 2 things that the 2 statistical analysis, which tells us the abundance of reads. And it is FPKM is used for paired end sequencing, and RPKM is used for single end sequencing. This is what we came to know. Finally, when we understood the difference between the FPKM and RPKM, we got to understand that it is an attempt to measure the absolute expression level. So, and finally, we could differentiate it from the read counts because it it is something that both FPKM and RPKM are far from read counts because read counts, they need to be normalized, and FPKM and RPKM values, because in read counts there are effects of gene length and sequencing depth that are present. So the longer the gene, the more hits it may get. So in order to remove that, it needs to be normalized. So that is why FPKM and RPKM values are the two things that we get. So finally, the final step that comes in the RNA-seq pipeline would be removal of all the genomic data that is without expression that is present. So in this, the crux of the matter would be to disapprove the null hypothesis that is present, and to remove the type 1 errors that we could have. The second step would be to have a quantile normalization and normalization process to take place. So it's a technique in which we map the original values of the corresponding quantile of a standard distribution. An estimation of sequence depth and experiment is determined by the upper quantile of the counts. So this is what we understood from it, that comparing the quantile values instead of the raw values will eliminate the difference. So we understood that there is a difference that could arise from technical variations such as the amount of RNA that was sequenced or sequencing of machine type, and some unexplained differences as well. But quantile normalization is used to remove all of that. And finally, we also understood that there are we can also use the log values to use and transcribe it, these quantile normalized values. It is much easier in approach and gives much better results when we use log values. And finally, we came across the machine learning approaches that were the PC analysis and factor regression analysis, particularly talking about the PC analysis because it's a very important subject for anyone who's trying to do this. It's going to tell you something very preliminary, and it's going to be very intriguing for you if you are doing PCA for the very first time. So PCA analysis is something that compresses the expression data that you had from thousands of genes into a single one-dot graph. And if you had a look at the TBio info server, there's a very there are many pipelines that you can run for the PC analysis. One is the unsupervised. The second one option you get is in the utility section. So that is how you can run it in a very simple way in the using the TBio info server. So it does not select some characteristics and discard the others, but instead it constructs new characteristics based on the whole transcriptome data set that you provide to the system. And then it gives us an accurate summary of all the variables that are being studied. So it tells us what you have done in a best possible way that it could be understood, and so that you can get the expression values. It's basically to explore what you have just done. So, and the result that comes out is that the genes with the largest variation between the samples will have the most influence on the principal components. So you will see there in the principal component analysis that you will receive the result in forms of percentage. So that percentage will tell you how much variation is present, and the largest variation can be used to study further your data. So bottom line that we understood in PCA is from Omixlearn, that was the samples of similar gene expression when clustered together in the PCA plot. So this is something that you can look for when you are doing your preliminary studies, that the samples that you put in, if they are clustering together, then that is what the PCA plot is going to show you, and you can take your studies ahead with that. And so this is what we understood. And the second one was the factor regression analysis that happens. So factor regression analysis was the method to perform so that the experimental designs that include two or more factors such as environment or time, they can be studied. The result from the factor analysis allows us to study a number of factors and their interactions instead of when we are limited to the differential expression, which only allows comparison of at least two experimental factors. So this is how it really helps us. And finally, we came to a point of biological interpretation. I'm still yet to perform this and do this, but from my understanding, what I understood that the genes that we get from the PCA analysis, we have to analyze them, study them. Why are there outliers present in the PCA plot? We study more about them. We use the David software that is present, and we can use the ensemble for finding the ensemble IDs if your database has ensemble IDs. So that is the step of biological interpretation that happens to be the last one. And finally, coming to why I chose prostate cancer, because the first thing that came to my mind and in which all the other mentors also helped me very much is that, first of all, it's an age-related disease. And that's why my grandfather also suffered with it. So that's why I was very well keen to understand it, what are the things that it holds. And my first question then came with the age relation of this cancer. Why is it that in that particular age or in some particular age, some biomarkers are, some genes are overexpressed, which result in this cancer? And further on, increasing the Gleason score and making it a very malignant cancer, and of course, being a life-threatening cancer that it is. So from this, first of all, I did a database search. I used, if I take you to the data set, I did a database search, and from the mentors, Dr. Harpreet ma'am told me to search from Firebrowse geo datasets. I liked the Firebrowse very much, and I took the dataset from it. And I also understood that there's a plot present in Firebrowse itself, which tells you about how your data is, your quality of data, and what visualizations can be made from that data using the plots available there as only. So from Firebrowse, I took the data sets for prostate cancer, adenocarcinoma prostate cancer, and I took the data sets. And what I really wanted to understand was the age and the relation of age with the overexperienced or genes that were present, and identification of clinical markers in prevalence of prostate cancer in age males. That is going to be my research question, my preliminary research question. Finally, when I get through this, I want to understand more about it. So all these factors I got from the dataset are present in my dataset. And what other factors can be used to understand the expression levels of genes? For example, race, ethnicity, Gleason score if possible, and the PSA values. PSA values, PSA is the test that is used for detection of prostate cancer. And that is the preliminary test that is used for understanding prostate cancer. There are, however, studies being done to have much better screening tests done than PSA tests. And finally, the next step are going to be, I have got my data set. The next question that I want to ask all of you is, first of all, the groups that I should separate my data sets in. The answer that I received today was to separate it into groups of ages separated from 10 years, like 50 to 60 years of age, how many samples are present there, then 60 to 70 and 70 to 80. And finally, the next question that I would like to ask all of you is that, should I separate it into cancer types, non-cancer type, and what all things that can be derived from this, and what all studies can be done, and your respective comments on it as well. So with that, I would like to end this preliminary presentation. And yes, this is the PCA plot that I got from my data set. This is a very, very preliminary and primary thing that I did with just about 25 to 30 samples. And this is what I received. So if you can shed a light on this as well to help me understand, or if you can tell me about this as well. So this was my presentation, and thank you all for being very patient listeners. And thank you for listening to me. Over to Sonalika ma'am.
Thank you so much, Hriday, for explaining your progress in such a wonderful way. I would want Dr. Harpreet to please guide you with the questions you have put up. Over to you, Dr. Harpreet.
Thank you, Sonalika. Thank you, Hriday. Okay, so actually, yeah, I mean, it's nice presentation. Okay, so because you just have prepared your data, so I mean, still you need to exactly name your samples based on their groups, so that you will able to get at least exploratory, first perform exploratory data analysis, that is PCA, to get an idea like what kind of questions can be addressed, right? And maybe right now you have taken just, I guess, 20 to 30 samples. Just do it with all the samples, so that you will get idea like what kind of pattern within your whole data, so that you can actually then next you can address your questions.
Okay, ma'am. Sure.
Okay, so instead of considering, and of course, instead of the sample ID, you also need to define, I think I have also already told you, like you need to insert another group. So another row that is actually explaining like what are these groups. So then based on those groups, just perform your PCA. So then based on the exploratory data analysis, then we can understand like what are the different questions that we can drive from this, right?
Yes, ma'am. Okay. Understood, ma'am.
Okay. Okay, thank you, Hriday.
Okay, ma'am. Thank you,
So
ma'am.
maybe I can maybe I can add to this. So one thing that you are doing is exploration of the data. I think in parallel you should be doing literature review to see, one, the publication that came out with the prostate cancer repository on NCBI. So, or sorry, on TCGA. So every project that came out on TCGA comes with a paper, and that paper describes what did they do with that data. So you're looking just at gene expression data, but a lot of the objectives from TCGA was to understand the integration between different types of data. They developed some methods. Some of those methods are also available on the platform, but they provide you with a sense of what did they find? What did the experts in the field that collected and analyzed this data come out with, right? And that will give you some clear idea about some of the things that should be a confident way to proceed if you want to, build on what has already been done. The second part of that are some most current literature, like on systematic review, really up-to-date systematic review, or even just a review paper that will give you some sense of what are the open questions about prostate cancer. I know from a little bit of background that, I have in participating in projects on prostate cancer, that there's a specific drug resistance issue that occurs at later stages of prostate cancer called castrate-resistant prostate cancer, and that's a big topic of research right now. So when you think about precision treatment and therapies that are being developed now, a lot of them are being developed for the cases that do not respond well to treatment. And so maybe that's one thing that you can look into. Maybe there's some issue with early stages where early detection of that resistance can guide the clinician to find an appropriate treatment. Maybe it's drug repurposing. If you want to treat cancer, but you don't have any new ideas, any new drugs, maybe some existing drugs that are being used for something else could be applicable here, but you need to know which patient population. So those are some of the questions that, I know are being explored, and you should consider how does that fit in with what you have. The other thing that I definitely agree with Dr. Harpreet about annotating these by some kind of phenotype. And so what are the phenotypes that you have? You have age, you have maybe ethnicity, you have maybe other treatments that are already, right? Yeah, so all of these. And so the question is, out of all of these, what types are categorical data, like race or ethnicity would be categories, and what of them could be converted into continuous data? So for example, continuous data could be survival, or they could be maybe you can turn some treatment into continuous data, or some kind of numeric data that could be used in more big data analysis, right? Where you could maybe find correlation between different variables. So I think those are some interesting questions. So when you do this kind of exploratory analysis, one, do literature review together with data exploration. Number two, when you explore your data, look at the data itself, gene expression, but also look at all of these phenotypes and identify what types of variables are these and what you can do with them. And then finally, PCA is a good way to get a sense of what's going on, but it's not the only way. And so another way that I would suggest, and this is something you should include, are some maybe box and whisker plots, so box plots. They tell you the distribution and some outliers in your data. So maybe, and even in PCA, you could see it, you do have some outliers. Why are these outliers there? Are they different because they just, overall the expression is lower? You will see the median lower. Yeah, like for example, in the one on the left, you definitely have an outlier right there, right? But before just removing that outlier, you also can ask the question, why is it there? What's unique about that sample? Now, the other thing to consider here is that all cancer data sets are very, very heterogeneous. So they have a lot of variability in them because these are real people, coming from different places, being sequenced at different places, right? So another, I think, question that you might ask here is essentially, like Harpreet said, do I just look at a subset or do I try to look at everything? And within all of the data that I have, what are some of the major patterns that I see? So maybe these could be helpful for you.
Perfect.
Thank you so much.
Thank you so much, Ilya sir. Sir, if I could ask, and Harpreet ma'am also, that from the TCGA papers that I found this, I was unable to find the research that was presented. If maybe any of you guys can help out to understand and search the papers, that would be really, really helpful. I would really appreciate.
Sure,
Thank you.
we'll share the papers with you.
Actually, that's a really good point. We do have a new course that we're going to publish either today or tomorrow, and that's going to be all about the project stages. And one of those stages is literature review, how to find literature, how to find data sets. So Dr. Kaur made a very great, nice tutorial about that. So we'll share that with you once it's done.
Definitely. Thank you so much, sir. And thank you so much for with that, I would like to pass on to Sonalika ma'am. Over to you, ma'am.
Thank you, Hriday. So Akansha, can we have your presentation now? Akansha is one of our research fellows from Amity University, and she's pursuing a bachelor's in biotechnology. Over to you, Akansha. You may share your screen now. Thank you.
Thank you, Sonalika ma'am. Is it visible to you all?
Yes, your screen is visible.
Okay. So currently I'm working on glioblastoma, and since 1 month I joined from last 1 month, and I'm, as you introduced, as Sonalika ma'am has introduced, I'm from Amity University, India. And so why did I choose glioblastoma is most probably because it has most aggressive primary brain tumor, and it has high mortality rate. Other than that, it has a hindrance, blood-brain barrier, which leads to failure of drugs to target the glioblastoma cell tissues. And the real cause of glioblastoma is still unknown to us. And the dataset for the research I chose is from GEO datasets, and it has 3 subtypes of samples. Total samples are 118, and it contains adult glioblastoma, neurostem cells, and lower grade glioblastoma. Other than that, it has metadata kind of tissue types, patient age, patient gender, IDH status, and based on IDH status, I proceed my research questions. So my research questions are basically to demonstrate the drug targets based on the differential expressed genes. Before that, I status of glioblastoma and how these genes vary in IDH wild type and IDH mutated tumors. So the current workflow I am following is
I have I pre-processed my samples into normal stem cells, glioblastoma cells, and lower grade glioma. And further, I performed scatter plot uh, on glioblastoma and normal stem cells. This is what I got results today based on my PC1 and PC2 result. I performed scatter plot. So these are the normal tissues, and these are IDH1 mutated type, and blue one are the IDH1 wild type. So I still I want to know how to proceed further on after the scatter plot. So this is all about my project.
Thank you so much, Akansha, for sharing your progress, and we would be looking forward to more of your work. And Dr. Harpreet, do you want to add something?
Yes. Thank you, Akanksha. Can you just go over your PCA plot? Scatter plot, PCA scatter plot. Okay, so here you have kind of, we can see, I mean, clearly see the difference between. Again, if I'm just looking at the, what are the blue samples that are wild type?
IDH, yes, ma'am, wild type.
Okay, so again, if you look at this wild type sample, you are like still kind of three groups, if I'm not wrong. One is completely on the left side, so other one on the right side, and some of the samples are in between. Can you just actually label these samples? Exactly what are these samples? Similarly, based on the mutations, I mean, mutated one, I think the gray samples. And again, there are like kind of three groups, but still I'm not considering the third as a group. So can you label these samples further with their other phenotypes? Actually, like what are the other phenotypes in your data? Probably because these are the three different groups, we need to understand why they are like making a three different clusters.
Yes, so
So you
based
have
on
the phenotype, like already, I mean, you have maybe, you need to explore like what kind of, already metadata you have. Maybe based on that, you can label these samples apart from these classes.
Okay,
Because you need to
I have
understand
to.
why they are making three different groups first.
Okay, you mean I have to consider other metadata as well?
Exactly. Just to understand first, what are these three different groups, right? Why it's not making a single cluster. So probably there must be some other phenotype.
Okay.
That is,
Yeah, sure.
do
So
that
I
thing.
have gender. So based on gender also, I can proceed further.
Okay, so you just need to explore your metadata and then just label your samples based on different types of the metadata, right? Like here you just label your sample based on this part, these three phenotypes. So you must have other phenotypes. Just try to do that as well. Considering
Okay,
these
sure.
as it is like normal. For example, I'm just considering normal male or female. So similarly, maybe with the treated or untreated some drug, I mean, if you are considering wild type, so that is something that we need to understand before proceeding further.
Okay, ma'am. I'll look into it.
Okay, thank you, Akanksha.
Thank you, ma'am.
And Ilya, if you want to add something.
Yeah, so I would say again, similar response to Frida. So you, a lot of times it's great to jump into the data and see what's available, but, take a look at the literature. There is a lot that people have tried. And so if we just learn from our own mistakes, we're essentially going to be wasting a lot of time because we're going to be trying things and they might work, they might not work. You might have findings that seem interesting to you, but somebody already did exactly that, right? So before you waste a lot of time, use your time to understand what has been done, how it has been done. It will give you also some ideas. Again, just even take a look at what everybody presented today. There are some standard methods that everybody will use. PCA, differential gene expression. There are other methods out there, and there are other questions out there beyond just what are the differentially expressed genes. These are popular methods. You can try them. They could be very effective, but the overall design will be much more interesting if you're more informed. What else could you do? How can you do this differently? How can you not repeat what somebody else did? That will also make your project stand out.
Yes, so I look into it. I have actually less time because of my university project. So I'm proceeding with this as of now, but I'll definitely look for the other questions as well.
Yeah, which is absolutely fine. But again, remember, this is a research fellowship program. You can do for your university whatever your university requires, but we make this program available to those that are interested in the research question. You know, if it's just an assignment, that's fine. You can treat it as an assignment. For these discussions specifically, these are places where we discuss what you understand in order to be asking questions that are more interesting, more specific, and are not just there because somebody told you. You know, these questions arise as a result of something that really spikes your interest. So I think, you know, that's just something that is specific to this program. There are many other programs that we work on that are more training programs.
Yes, sir. Okay, sir, I'll look into it.
Thank you, Ilya and Dr. Mohit, for your suggestions, and thank you, Akansha, for sharing your progress with us. So for those who are a part of the LBRN Research Fellowship Program, we would be having a meeting with you at 3 p.m. CST, and that would be in few hours. And so we have Abubkar here from the LBRN group. I hope you have received the invitation, and you can meet Ilya there and share your queries. Okay. Do you have any other questions, Abubkar? You can ask me in the chat box. And I also wanted to introduce you all our upcoming program that is on precision medicine. It would be starting on July 6th and would end on August 5th. It's a month-long training program. And we have a free webinar to discuss the role of bioinformatics in precision medicine on 2nd of July. I invite you all to be a part of our webinar there and learn more about how bioinformatics could be used for research in precision medicine sector. And I would be sharing the program page with a link with all of you as well. Here is the link. You can go through it. Here we have mentioned all the session details and also what all would be covered on what all dates. And you can also go through the registration process for the same. Now, those who are a part of the research fellowship program, they would get the access to our programs with 50% off on the program. So that is what you get at being a part of the research fellowship program. Those who are interested may mail me at marketing@fine.bio. And I would be there to assist you with things in a more further way. And also talking about the weekly progress report. So we expect all our research fellows to fill out this weekly progress report. And those who have joined today, we would be having another batch meetings with you. And so what are these batch meetings? We have weekly research fellowship meetings where we discuss all the we have presentations by our research fellows and we look at their progress who have almost completed their projects or are on the verge of doing so. But we take the progress report and also help out each and every research fellow at different stages in their research project in batch meetings. So we have divided our research fellows in 3 batches, and we would have the batch 3 meeting scheduled for tomorrow. And people who are a part of the LBRN Research Fellowship meeting, you all would be having a meeting that is scheduled on the calendar. And I've already sent out you the invite, so you would be updated by that. And also we would be soon uploading a calendar on our portal so that you stay updated with everything. And we expect our research fellows to fill out the weekly progress report so that we know about at what stage they are at and if they are facing any sort of issues, and then we can help them proceed further. And also, I would want you all to please update your profiles, though many of you have already updated, but those who have not, it's for them. Please update your profile, add a profile, picture to your account, as Ilya already told, and also link in your social accounts so that it's easier for us to feature your profile with your performance on our social platforms. And I was just having a word with Abubkar, and he was asking me how he proceeds further. And also, Yashas Ravi, you had few doubts about the research fellowship program, how it works. So the thing is that in the first 15 to 20 days, let's say, or you can take a month as well to complete the course material. And then you have to submit your research proposal. And in your research proposal, you have to address your problem statement, your project summary, and link for raw data, and also the reference publications which you went through. So in the first month, you would have to go through the asynchronous course material, the project samples we have to understand how the analysis could be done, and then go through the literature part as well. And as Ilya told, that we would be helping you with courses on how to review literature in a more better way, right? And then you come up with your own research question. You have to submit your research proposal form to us, and then our mentors would guide you with the research proposal which you submit. It could be modified by our mentors as well to refine it in a much better way. Your question could be refined at each and every level, but our mentors would be there to guide you from selecting your research question till completing it. And I'm so sorry. Yes. So once Yashas Ravi, I feel you are interested to know further, I would be mailing you all the details, and then we can proceed further with your enrollment. And does anyone has any doubts, you can put it in the chat box so that I can answer it for you. And if not right now, then you can mail me at marketing@pine.bio. I've already shared my mail ID. And those who are looking forward to apply to the research fellowship program, I'm also sharing the link of the research fellowship form with you all. You can proceed further here. Here is the link. Okay, thank you everyone for joining us today. We would be looking forward to your progress reports, and let's meet batch 3 members tomorrow and discuss their report. And also we have changed the time for the batch 3 members, because we were noticing that many of you were not available at that time period. So I've sent you out the invitation, and the calendar has the time on it. So please stay updated. Thank you so much everyone for joining. Bye-bye.
Thank you, everyone.
Thank you.